So one of the really exciting themes in hematologic oncology right now is the ability to selectively inhibit important pathways for cellular biology. And as we have developed tools to selectively inhibit those pathways, the theme right now is to combine agents together so that we can both dampen down on parallel pathways for cell growth or to inhibit mechanisms of resistance. Sertolatinib, which is a dual inhibitor of both the SYK pathway as well as the JAK pathway, is able to accomplish this with a single molecule. And today, on behalf of my colleagues, I'll be giving you an update on an abstract that will be presented on Sunday in the morning, and the full data will be seen at that time. So the purpose of this research, which is a phase 2 multi-center open-label study, was to investigate an oral medication. It's a competitive ATP inhibitor, as we mentioned, of SYK and JAK. The name of the drug is sertolatinib, and it inhibits two different pathways that lymphoma rely on for survival. The pathways that this specifically targets include JAK1 and 3, SYK, and TYK2. And the purpose of the research was to determine not only the safety of the molecule, but also its effectiveness in specific subgroups of non-Hodgkin lymphoma. So this phase 2 study proceeded after an initial phase 1 first-in-human study had identified safety and dosing. It was an open-label study, and it accrued patients who had relapsed and refractory CLL and SLL, follicular and indolent NHL, and a T-cell lymphoma cohort that was just opened based on work done by one of my colleagues when he was at the Mayo Clinic, looking at aberrant expression of SYK in T-cell lymphomas. Sertolatinib is an oral medication. It's taken twice daily at a dose of 30 milligrams in the current phase 2 study. And it's continued until either progression of disease or toxicity. What we've been able to demonstrate with the results of the research is that at a dose of 30 milligrams BID, sertolatinib inhibited the pathways of interest. So both SYK and JAK are inhibited at maximal amounts at physiologically obtained doses at 30 milligrams BID. The study initially had accrued patients at 35 milligrams BID, and that dose was then adjusted to 30 BID based on analysis of the pharmacokinetics and safety profile. And I'm happy to say that that adjustment has reduced some of the incidence of side effects that we saw and has maintained effectiveness. And the effectiveness that we've seen overall in the cohorts is that 50% of patients, out of a total 47 accrued to date, have had a response. They've predominantly been partial remissions with a singular CR at this time point. And of note, in CLL and SLL, where we know that the B-cell receptor pathway is important for the survival of the tumor cells, the response rate is 67%. As with other agents that target the B-cell receptor pathway, we do see lymphocytosis and elevation in the lymphocytes. In follicular lymphoma, the response is 56%, so 5 of 9 patients. These responses are brisk. Patients feel better quickly. After just 2 cycles, we're seeing improvement. And as we mentioned, they're predominantly partial remissions, but they're sustainable. And so at this time point, just about 50% of the patients remain on study drug, and we have 4 patients who are approaching 1 year with disease control and good tolerability. The most frequent side effects that we've seen are fatigue, diarrhea, which occurs early and responds well to a dose reduction in anti-motility agents, nausea, and some cytopenias. So in conclusion, sertolatinib is an orally available dual inhibitor of 2 pathways, both SYK and JAK, in a single molecule. We've seen consistent activity in relapsed and refractory CLL, SLL, and follicular lymphoma, with notable responses of 67% in CLL, SLL, and this cohort continues to accrue, as well as in follicular lymphoma with 5 of 9 partial remissions for 56%, and again, continuing to accrue. The aphorism that it's always good to be the first patient on a clinical trial cohort remained true. Our first T-cell lymphoma patient achieved a complete remission, and I think is proof of principle that the biology suggesting that SYK is aberrantly expressed in T-cell lymphomas and may have an important role in its pathogenesis, what holds true. Responses, importantly, have continued to improve over time. with 7 out of 9 patients having further reduction in their nodal mass on their second and third scans as they continued on study drug. And we expect by year end to have additional information on the 30 milligram BID cohorts in SLL, CLL, follicular and indolent lymphoma, and T-cell lymphoma. And with that, let me conclude and take any questions. Thank you.